Hey guys, how are you? Today we're going to talk about protein synthesis. So here's your assignment for today. So it's log on and take the quiz labeled Biology Quiz 19, DNA, Structure, Function, and Replication. Okay, so make sure you are taking that quiz today on March 26th. Okay, so make sure you do that. Before you do any of this, go to Kia and take the quiz labeled Biology Quiz 19, DNA, Structure, and Function, and that will entail any information that you did for lesson 1 and 2 from the 19th and the 23rd. Okay. Then after you're done with that, come back here, watch this video. Obviously, that's what you're doing. And then you want to complete this worksheet, okay, and have that in by tonight. And you also want to write your summary for the day and have that posted with the worksheet as well. All right. So without further ado, I think we should get into it. So protein synthesis, if we go with some of the words here, the main function of DNA is to hold genetic information. We learned that in the past two lessons. But why, right? Why does DNA hold genetic information? What does DNA actually do? Does DNA actually have a function? And I'm going to be using this analogy of a cookbook, okay? And some of you might hate it. Some of you might love it. I really like it, okay? So a cookbook is nothing more than a collection of recipes. And that is exactly what your DNA is. Okay, your DNA is a collection of recipes, but not for food, obviously, but for DNA, for proteins, rather. Okay, so I'm going to be going through this analogy, right? So this analogy is going to be, let me grab my pen here. Okay, so the DNA and a cookbook. Okay, the DNA holds all the information for all the proteins that make you, that give you your traits, right? So that's what we're cooking. We're cooking proteins, but those proteins are responsible for traits. In a cookbook, we're making some type of meal, right? So the meal is the goal, right? The meal is the goal. And for DNA, what's the goal? The goal is proteins. Okay, we want to make proteins. So you look at DNA to get the instructions for the production of a protein, just like you look at a cookbook for the instructions on how to make a meal. So I'm gonna make a meal up just for argument's sake, okay? And I'm also going to change my pen so that we can see it a little bit brighter if I have to go into this gray area. So we're going to say beef Wellington. I don't know if you guys ever heard of beef Wellington. It's a British dish made famous by Gordon Ramsay, one of my favorite guys on the planet. And so that's going to be like our little example. So we're going to look in a cookbook. We do not know how to make beef Wellington. So we are going to look in the cookbook to get the recipe. We're going to follow those instructions, and we're going to make beef Wellington. Same thing with DNA. We want to make a protein. Whatever that protein is, let's say it's an antibody, okay? We can go back here and put antibody. An antibody is a protein that helps fight off infection, okay? So we need to look at the instruction manual on how to make that antibody, just like we need to look at the cookbook to make the beef Wellington. Okay, so let's go back here. Okay, so if we take a look at this little picture here, this is a very simplified kind of diagram that shows you how a protein is made. Okay, so let's go through this picture on here real quick. Right, so here's a cell. This whole thing is a cell, right? That's our cell. And inside the cell, you have a nucleus, which is going to hold our cookbook. Okay, it's going to hold our cookbook, which is our DNA. DNA, and I'm just going to quickly go through this in a very quick, like kind of Cliff Notes version. DNA is the entirety of the recipes for all the proteins in your body. Okay. Just like our cookbook has, or actually I can do it here, okay? Our cookbook has all the recipes for everything imaginable on the planet Earth, right? So this cookbook has all the desserts in it. This cookbook has all the entrees. It has all the appetizers. It has all the cocktails. It has every possible thing that you could think of being cooked is held in this cookbook. It's the world's biggest cookbook. It has every single recipe ever in it. Just like this DNA, this cookbook has every single protein that could be made for you, right? So your DNA is different than your parents' DNA or different than your siblings or your classmates. So their cookbooks are all unique to each other, right? So your cookbook holds all of your protein codes, and that's what this is, right? This is a recipe code book. That's all it is. It's a book for codes to make recipes. And that's all DNA is. DNA is nothing more than a reference guide, right? The cookbook itself doesn't make the meal. The cookbook doesn't make the beef Wellington. The cookbook is going to be the reference for something else to make the protein, just like your DNA, okay? It's for the reference for something to make the meal. Your DNA is a reference guide to make a protein. The DNA itself doesn't make the protein. Just like the cookbook itself does not make the meal, right? There's a chef that makes the meal. And over here, we're going to learn who the chef is and all that other stuff, right? So all the recipes. So this cookbook has all the recipes, right? All recipes, okay? But we want to make beef Wellington, right? We don't need all the recipes to make beef Wellington, right? We need one recipe, and that one recipe is the beef Wellington recipe, right? So we're not going to look at all the recipes. We're just going to look at the one recipe that deals with the beef Wellington, okay? And we'll call that the BW recipe, right? Beef Wellington recipe, okay? Just like over here, we don't need every single recipe for every single protein in the body, right? Because this is our antibody. That's our protein that we want to make, right? And I'm going to signify that by writing AB, because that's how you write antibody, AB. Okay, so we don't need every single recipe because there are recipes here for insulin. There are recipes there for actin and myosin, which are in your muscles. There are recipes there for polymerase. There are recipes there for every type of protein in the body, but we don't need all of those. Just like we don't need the cookie recipes for the beef Wellington. We don't need the soup recipes for beef Wellington. We don't need all those recipes. We only want the one recipe for the antibody, and that's what this is, okay? And those individual recipes we're going to see are called RNA, and or come in the form of RNA, which we will see more in a second. Okay, so that's equivalent. So the DNA is equivalent to the cookbook. The RNA is equivalent to the individual recipe that we want, and then that recipe is going to be read by somebody, okay, or processed. Okay, read or processed by somebody. And over here, it's going to be read by the chef. Okay, the chef is going to read the recipe, and then the chef is going to produce beef Wellington, right? Here in the cell, we don't have a chef. We have something called ribosomes. Ribosomes read the recipe, and they produce the protein. Okay, so we have the cookbook, the recipe for the thing that we want to make, the person or the thing that's going to read the recipe and then produce the meal/protein. Okay, that's why I really, really like this analogy. And this whole entire cell, I guess, could be the restaurant that this is all going down in. Okay, if that makes any sense. All right, so what we want to do is we want to take this step by step because there are two things that we want to learn about today. There's something called transcription, and there's something called translation. And we need to know what these two things are in order to understand protein synthesis. So transcription and translation are two things that we want to get to today. All right, so let me erase all of this and then we'll get to it. Okay, so the best way for me to do this is to go to this picture. Okay, this is one of my favorite pictures, and I'm going to zoom out a little bit so we can see more of the picture here, okay? This is one of my favorite pictures to use for this because it tells you so much in such a simple picture. So what we have here is we're looking at a portion of the inside of a cell. Here's the nucleus, okay, where the DNA is held, and I'll draw the DNA here. Okay, that's my DNA. Over here is my rough endoplasmic reticulum, the rough ER, right? Actually, let me put that in a place you can actually see it. That might be more helpful, right? Okay, so this is the rough ER, rough endoplasmic reticulum, and it's rough because it has ribosomes, right? And they're going to be very important to us, right? This is the Golgi body, right? Remember those. And what their job is to package and process proteins. Package, whoops, forgot the A. Package and process proteins. Okay, that's what the Golgi does. Okay, the Golgi apparatus. This little guy here, this little green guy with the little like starfish looking thing on top of him, that's our protein. Okay, so I'm going to go through this picture and show you how a protein is made. All right. So we're going to start off in the nucleus. So let's start off in the nucleus, and we can make the DNA blue. Let's make the DNA blue here. Okay. So this is the entire DNA. That's my cookbook. Okay. All the recipes are here in this cookbook. All right. And over here, we can draw the cookbook if you want. Okay, so let's draw the cookbook over here. Okay, so here's the cookbook. Now, this cookbook is very, very big. Okay, a very large cookbook. It's like the size of a small, like a smart car. It's very small because it has all the recipes for every thing ever produced, right? And we're going to say that this is held in a vault. And that vault is like at the basement of the Empire State Building or something, and it has this tiny little door. Okay. They put the book here, and then they built the building around it so that you can never take it out. So this is going to ensure that the book stays safe, right? And this building that it's in is the nucleus. Okay, it's my nucleus. So, and just like the nucleus, there's an opening so that things can go in and out, right? And we call those nuclear pores, right? We have nuclear pores that allow things to go in and out of the nucleus, just like we allow things to go in and out of here. The pores, however, they do not let the DNA leave, right? The DNA can't get out of those pores. The DNA is too big. And we don't want the DNA to get out. The DNA is really important, right? The DNA is the instruction manual to make all of our proteins, and our proteins are what make us us. Just like this recipe book is really important. We don't want the book to get out, right? So the book is way too big to get out the door, and that's the only entrance or exit to get in and out. So we want to keep this book safe in here, okay? So that's number one. So now, let's say that we want to make an antibody, right? So that antibody is a very small portion of the DNA. And we'll indicate that by putting a little red line. That little red line is the portion that is responsible for the instructions for the antibody that I want, right? There's my antibody, okay? Just like here in this book, this one little paragraph is the recipe for beef Wellington, right? That's my beef Wellington recipe, okay? I don't need the entirety of the book. This is, a million-page book with, a gazillion recipes in it. I only need this one recipe right there, okay? So here's the deal. The deal is that the ribosome, one of these ribosomes, is going to be responsible for making my protein, right? So one of these is going to be the chef. That's going to be Chef Gordon Ramsay. We're just going to use Gordon Ramsay because I really like Gordon Ramsay, okay? So Gordon Ramsay, who's my ribosome, we're going to put, we'll just write chef here, right? Chef Gordon Ramsay, Chef GR. He needs the recipe. Now, you might be saying to yourself, he's Gordon Ramsay. Why does he need a recipe? He should know how to cook this, okay? That's where I need you guys to not be so crazy and just go with me here on my analogy. Chef Gordon Ramsay is an amazing chef. However, he doesn't have the memory to remember all the recipes. So every time he wants to make something, he has to go into the vault and read the book and get the recipe out of the book. Okay, it's one of his very few faults that he has. Okay, just like the cell, every single time we want to make a protein, we have to go into the nucleus and look at the protein. The ribosomes do not know, or they cannot memorize. They do not have the capacity to memorize the instructions for all the proteins. So every time you make a protein, every time you drink milk, you produce something called lactase, which is an enzyme that breaks down lactose, right? If you're not lactose intolerant. And every time you drink milk, your body has to make those proteins, and those ribosomes have to look up the instructions to make lactase. So it's just the way it is. So now the chef here, this ribosome, the ribosome does not go into the nucleus and look for the recipe. He actually sends somebody else into the vault for him, right? And we call that a sous chef in the restaurant business. And I don't know how to spell sous chef, so I'm just going to write S-U-E-S. I think that's how you spell sous. Aurelian can tell me if I'm wrong or not. It might be E-U-S, but the sous chef, who's like the helper to the head chef, that sous chef is going to go into the vault and find the recipe and make a copy of that recipe. They're going to make a copy of the recipe. Same thing happens over here. The ribosome cannot go into the nucleus. So the ribosome sends something else into the nucleus. What gets sent into the nucleus is something called tRNA, lowercase t, R-N-A. Now, RNA is a version of, it's like DNA, but it's slightly different. Okay, DNA, okay, I'm going to write over here if you don't mind. DNA has nucleic acids called A, T, G, and C. RNA, however, doesn't have the same letters. It has almost the same letters. It has A, U, G, C. Okay, so RNA can't write the letter T, right? The sous chef he's a French guy. He can't write he doesn't know the English language as well as Chef Gordon Ramsay because he's British, right? No offense to French people. It's just going along with my analogy here. So every time that our sous chef, when he's copying the recipe, every time the sous chef sees a T in the recipe, instead of writing T, he's going to write a U instead to replace the T. And Chef Ramsay knows this, so it's not a big deal for Chef Ramsay. Okay, so tRNA, which is the sous chef, that's my sous chef. Okay, tRNA is my sous chef. The tRNA goes into, and we'll make him a different color. Okay, let's make him this turquoise color. This tRNA goes into the nucleus of the cell and reads the recipe for the protein that he is responsible to go in there and make a copy of, right? Just like this sous chef, Gordon Ramsay tells him, go in, get the recipe for beef Wellington and come back, right? That's what this tRNA is doing. This tRNA is going in there looking for the code or the recipe for this antibody, this specific antibody, and it has to bring it back to the ribosome, okay? So this copy that it makes, it's going to read the letters of the DNA, and it's going to make a copy. So let's go over here to my whiteboard, okay? Let's make believe that we are going to be looking at the DNA code, right? So here's my antibody code, okay, of my DNA antibody code. Let's say that code says this: A, T, T, C, 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 G, A, G, G, C, C. What is that? It has to be divisible by 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. C, C, A. Okay. So let's say this is the code of the DNA. This is the code this is the recipe for the antibody, just like this would be the recipe for the beef Wellington, so to speak. Okay. When the tRNA comes in, the tRNA is going to read this and is going to make a copy of this code. Okay. It's going to make a copy of this code. And you might be saying to yourself, why aren't there letters across from here, right? Why aren't we looking at, because DNA is a double helix, so there should be letters across from here. We only need to look at half of the DNA to get the code, right? Because we automatically know, because of Chargat's rules, that across from this would have been a T, across from this would have been an A if we were to draw a piece of DNA. So we only need to read half of the DNA. Okay, so when the RNA reads the code, it sees an A, it's going to make the complement to that A, which is not a T anymore, because RNA doesn't know how to read T's or how to write T's, it's going to write a U instead. Okay, so it sees an A, it makes a U. It sees a T, it makes an A, just like normal. Sees a T, it makes an A. Sees a C, it makes a G. 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 Sees a G, it makes a C. Sees an A, it makes a U. Sees an A, it makes a U. Sees a G, makes a C. Sees a G, makes a C. G, G, U. Okay, this is the copy of the recipe for beef Wellington. Okay, the copy of the recipe, the copy of the piece of DNA is called mRNA. And the M stands for messenger. Okay, the M stands for messenger. Messenger RNA. Okay, this is nothing more than a message, right? That's what this is. This copy of this DNA code is a message to give to the chef, right? And what made this? Okay, what made this piece of mRNA was the tRNA. The tRNA was the sous chef that went in and read these letters and made these letters. What does the T stand for? The T stands for transfer RNA. Okay, so this piece of transfer RNA read these letters and made the message that we call mRNA. And now this message has to go to the chef or to the ribosome, right? Okay, so let's go back to our reversal lesson. Okay, so over here, this green piece was my sous chef. Okay, I'm just going to erase my sous chef here for a second. Okay, and we're going to say that my piece of mRNA is this green portion here. So that's my messenger RNA. Okay, that's my message. Now the tRNA has to carry that message out back to the chef so that the chef can read the message. Okay, now one thing that I want to tell you is that the process of making this copy, okay, the process of making this piece of DNA into a piece of messenger RNA has a name, and that name is transcription. Okay, this process of DNA to messenger RNA is called transcription. Okay, that's what that is. Okay, so we've gotten one out of the two things. We want to do transcription and translation. This is the transcription part. We are transcribing, because that's like what monks did, right? They transcribed Bibles back in the day before the printing press. They would look at the code, they would look at one Bible and write it again as another Bible. Okay, and sometimes they would do that in different languages, kind of like we're doing it in the RNA language here. Right, so they're transcribing it. Okay, so we're going back to here. Okay, so this transcription that happened in here, okay, it made our messenger, and that message is going to the chef who's going to read that message, and then that message is going to be translated into a protein. Okay, so let's go back here. Okay, so let me just erase some of this stuff here. Okay, so let me erase this stuff. Okay, so this was called translation, and this is our mRNA. Okay, and this has to get brought, this mRNA gets brought to the ribosome. And that gets brought to the ribosome. Let me just take this out because I'm going to need this. Okay, so now what we're going to do, what the ribosome is going to do, okay, the ribosome is going to look at these letters, and the ribosome is actually going to make a protein by looking at the letters. Okay, so we see that I broke this up into 3 letter segments. Okay, these 3 letter segments, the reason I broke them up into 3 letter segments is because ribosomes can only read 3 letter segments. Okay, just like you read one word at a time. If you were to look at a recipe and it said, add 2 sticks of butter, you would read all those words individually. So each one of these sets of 3 is an instruction for the ribosome to read, and we call a a group of 3 letters in a piece of DNA or RNA, we call that a codon. So here we have 1, 2, 3, 4, 5 codons in this particular sequence. Okay, we understand that? Okay, so the ribosome is going to read each codon individually, and each codon is going to be the instructions for something called an amino acid. Okay, I hope we remember back in the molecular biology chapter, the macromolecule chapter, that all proteins are made of amino acids. Those are the building blocks for proteins. And you have 20 different amino acids, okay, in the human body. And the way that we make a protein is by stringing together these 20 amino acids in different amounts and in different orders, okay, because amino acids have different shapes. So let me bring up an amino acid chart. Okay, and we can see the differences in the different amino acid shapes. Okay, so here, let's see if this is a big enough chart. Okay, here we have an amino acid chart. Let me actually get a different one. Okay, this is a good one. Okay, this is a good amino acid chart. And if you look at this amino acid chart, you can see that these amino acids have different shapes attached to them, right? So this the only thing that differs on an amino acid is the highlighted parts. Okay, all the other parts are exactly the same. So the structure of an amino acid is always going to be the same, and we can get into that a little bit more later. But the part that varies on an amino acid is this highlighted part, and we call that the R group. Okay, and this R group changes from one amino acid to another. So glycine has a hydrogen as its R group. Okay, alanine has a CH3 as its R group. Valine has this structure as its R group. Tryptophan has this structure as its R group. And you can see that they're all different. Okay, and you do not have to memorize any of the names or any of the shapes of any of these amino acids at all. So please do not think you have to. But what I'm trying to show you here is that all of these are different shapes. So to make a protein, you're taking these and combining them together. And when you combine these together in different orders and in different sizes, you're going to get very different shapes when you make a protein. So for example, let's just make believe that glycine has a triangle shape, and that alanine has a circle shape, and that valine has a square shape, and that leucine has an asterisk shape, and isoleucine has one of these star shapes. Okay, if the code of a protein says triangle, triangle, circle, square, asterisk, that is the code, or that is the shape that the protein has to be in order to work. Then that's the shape the protein has to be to work. If we look at a key, it's very much like a lock and a key, right? If we have a key, okay, we have a key here, and that key has teeth on it, right? And those teeth are different shapes. Those teeth might be pointy and short. Some of those teeth might be rounded and fat. Some of those teeth might be, a little bit taller than the one next to it. This particular key fits into a particular lock, right? And if the teeth aren't correct on the key, then it won't fit, or it won't even turn the lock. You might even be able to get the key in, but you can't turn the key, right, if it's the wrong key. If this is the shape that the key needs to be, and I actually change the shape that this key is supposed to be, let's say I, instead of this being tall and rounded like that, let's say I point it off like that, that little difference in shape is going to cause this key not to work in this lock, okay? And the same thing goes for proteins, okay? If the code for the protein dictates this shape, if I alter this shape at all, then my protein's not going to work. And what that leads to is something that we call a mutation, and which leads to a disease, which is in the next lesson, okay? So it's very important that we follow the steps that are given to us in this code, right? So let's try to figure out how this code is actually used to make a protein. Okay, so I don't know if you guys have ever used a decoder ring. Okay, the decoder ring was something that kids played with back in the day, and basically it would be like a secret code where a certain number would equal a certain letter or something like that, and you would have to go back and forth between the code that you received and a little key that would tell you what the letter represented or what the number represented. We're going to use one of those here. Okay, and so let me wipe this clean. We're going to use this, and this is a codon chart. Okay, and this codon chart is going to help us to figure out what amino acids are being utilized in that code. Okay, and it's very easy to read. So you have three letters in a codon. So this codon says UAA. So when the ribosome reads UAA, it goes to the code and it says, okay, the first base is U. Okay, the first base is U. Okay, the second base was what? UAA. Okay, UA. So we know that the answer is in this box somewhere. Oh, did I really use a stop codon? Okay, we're going to go back here and we're going to change this A. Okay, because that's annoying. Let's just say this is now let's say this is a G. UAG, so that means that this had to be a C. Of course, I do that all the time, actually. I should learn my lesson. Okay, that should be a C. Okay, you all following with me? See what I did there? Okay, not too big of a deal. So now our codon says UAG. Okay, if we go back here, UA. Oh, I did it again. Wah, wah, wah. Okay, let's make believe it was a C. Okay, this and this are going to change. This is going to be a G, and this should be a C now. Okay, and I'll explain what I'm doing in a sec. Okay, so UAC, that's our codon. All right, so UAC. All roads lead to that amino acid, TYR, which is tryptophan. Again, you do not have to know what TYR means or what it represents, but you just have to know that that's the amino acid that is being coded for by that particular codon, right? So if we go back here, we can write underneath here TYR, and that is the amino acid that this codon told the ribosome to make. All right, so the ribosome read UAC. The ribosome went to the chart, UAC, and it said, okay, I need tryptophan, and that's what it made. Okay, and let's say tryptophan was a triangle shape. Okay, and then the ribosome reads the next one, GGG. So it goes back. It says G, 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 right? G, 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 and they all meet up right here for glycine. So GLY. So GLY. And let's say that is a circle shape. Okay, then it says GCU. So the ribosome reads GCU and it goes to GCU. GCU. And that leads us to alanine. Okay, ALA. Okay, go back here. ALA. And let's say that is a square shape. And then UCC. Go back here. UCC. Serine. And you might be asking yourself, why are there doubles and triplets and all this other stuff? The reason is because we are set at 20 amino acids. We only have 20, but we have 4 letters, right? U, C, A, and G. Okay, and you can put those letters together in 64 different combinations. So that's why we have doubles. Okay, we can only make 20 amino acids, so we have to have doubles somewhere. So that made serine, okay, which was UCC. Okay, UCC made serine. I'm on a little bit of a delay here, serine, and we'll say that is an asterisk shape. Okay, and then the last one is GGU, and we go back here, GGU, GGU, and again that's glycine. That made glycine again. Okay, so we go over here, and that's GLY, and that gives us a circle. So this is the protein that was made from this code. right? So this initial DNA code gave us this copy that the sous chef made for the chef. And then that copy, that message, was given to the chef, and the chef read that copy and made these amino acids in the order in which he was supposed to make them. You can't change these. If you put this GGU and you switch it with the UCC, that makes this circle go over here and the asterisk move over here, and that's not going to give us the right protein. Just like if you did that with a key, if you took one of the teeth and switched it with another tooth of a different size, that key is not going to work, right? So this is our finished protein. That's our amino acid. That's our antibody, rather. Okay. This process, which we call transcription, was the process of taking DNA and making it into RNA. The process of taking RNA and making it into a protein, because that's what this is. This is now a protein, right? That process is called translation. Okay, that process is called translation. So the process of going from DNA to RNA, transcription. The process of going from RNA, mRNA, to protein is translation. Okay, so the chef has translated, or the ribosome has translated this message into the actual meal. So this is beef Wellington. All right, that's my beef Wellington. Okay, right here. Now, if we go back to our, let me erase this real quick. Okay, if we go back to our versal lesson, the same thing is happening. Okay, we go back down to this picture. Okay, so let me zoom out a little bit. Okay, there we go. Okay, if I zoom out a little bit. Okay, so our nucleus was here. Okay, a little message was produced by the tRNA given to the ribosome. Okay, so translation happened in the nucleus. The ribosome made the protein. Here it is. Here's our protein. Okay, there's our protein right there. That little green guy. Right, now this little green guy is going to, it's not done yet, right? The protein's made, but we need this protein to do its job. Now, our protein was an antibody, right? And antibodies typically will help fight infection. And infection is typically outside the cell. So this protein needs to be sent somewhere, okay? Now, typically proteins are sent in one of three places, and we're going to see that in a minute. But in order for this to get sent anywhere, it has to go to the Golgi, and that's what it's doing here, okay? This protein is being put into a little vesicle, okay, and that vesicle is made of phospholipids, just like the walls of the Golgi are made of phospholipids, and just like the walls of the ER are made of phospholipids. This vesicle is going to take this protein and give it to the Golgi. So here's our protein in the Golgi. Now, what I said before, the Golgi is going to help package and process proteins. All right, so let's take a look at those words. Package. Okay, and what do we mean by package? Okay, think of it as a UPS or a postal service or a FedEx place. We're going to put that protein in a package, in a box, and we're going to ship it to where it needs to go, okay? And that shipping is the processing, okay? The packaging is putting it in the proper container, and processing is sending it to where it needs to go. So where does a protein typically need to go? A protein can go one of three places, okay? A protein can go outside the cell, right, which is typically to the membrane, right? In order to get out of the cell, it needs to go to the membrane, okay, which is the outer exterior portion of the cell. It could go back to the nucleus, right? Maybe the DNA. So this protein can leave the cell. This protein can go back to the nucleus. Maybe the nucleus needs a protein like helicase, or maybe the DNA needs the protein polymerase to make copies of itself. You don't know. Or that protein can be sent somewhere in the cytoplasm. Maybe this protein goes to a mitochondria in the cytoplasm. Okay, maybe it gets used by a different organelle in the cytoplasm. But those are the only three places it can go. Okay, the Golgi is going to put it into the right box, just like the UPS is going to put it in a box and then slap a label on it, and that label gives the address to where that box is supposed to go. Right, that label will say, 555, West 25th Street or whatever it is. Okay, and then it gets processed and gets sent to where it needs to go. That's what's happening in the Golgi. So this little, you see, let me just erase some of this stuff here. You see like a little purple thing being attached to the end of this protein. That little purple thing at the end of the protein, which was not present here, okay, that little purple thing is the like the shipping label of where it's supposed to go. So this protein is going to move through the Golgi and it's going to get processed. It's going to get packaged and processed, and then it's going to get, once it reaches the edge, it's going to get put in another vesicle, another transport vesicle, and it's going to get sent to where it needs to go. Again, either outside the nucleus or the cytoplasm. So in this case, it's leaving the cell, okay, because it's an antibody and it's going to kill little bacteria over here, or it's going to help try to kill that little bacteria so that it doesn't make you sick. Okay, so just to kind of recap what we talked about, okay, we wanted to learn about protein synthesis, the making of proteins, right? That's what protein synthesis means, to make. Synthesis means to make proteins. That's what DNA is. DNA is the collection of instructions on how to make these proteins, and that's all it really is. It's like a cookbook. So in order to make a protein, we have to reference the cookbook, we have to reference the DNA, which is held in the nucleus, and we have to make copies of the instructions for the particular protein that we want to produce. And that copy is called mRNA. We can even put that over here, mRNA, okay? And that's done by tRNA, that is made by tRNA, okay? And RNA has a U instead of a T, okay? U, no T's, okay? The DNA is the collection of those proteins. The mRNA is the copy of the protein that we need, okay? Copy of the instructions for the protein that we need. And then the ribosome is going to, so this was called transcription. Don't forget that. Transcription. Okay, and then that copy of the RNA gets looked at and read by the ribosome and gets made into our protein, and that process, which is done by the ribosome, is called translation. Okay, and the next class that we're going to have is going to deal with how does this get messed up? Okay, how does this key come out wrong, right? How does this protein come out wrong? Because if that protein is supposed to be this shape, right? If the protein has to be this shape in order to work, and it comes out instead like this, okay, this is not going to work, okay? And a good example of this is something called cystic fibrosis. Cystic fibrosis is a disease that doesn't allow your lungs to clean themselves, and it's done by a protein. There's a protein that allows your lungs, allows water to pass through your lungs in order to clean the mucus that's in your lungs naturally. Okay, if your protein, if your cystic fibrosis protein is not the right shape, you do not have water able to pass through your lungs. It does not clean the mucus, and you can die before the age of 30. Okay, that's how important the shapes are. Okay, so we want to learn in the next lesson about mutations, and a mutation is when the letters either in the DNA are wrong, or maybe they're wrong in the copy. Maybe the ribosome read the codons wrong, and the protein came out with the wrong shape. And if you come out with the wrong shape, that typically means you have some type of disease that you do not want. Okay, so with that, okay, we should know what transcription is. We should know what translation is. Okay, transcription, DNA to RNA. Translation, RNA to protein, you have to use this chart, and that chart will be given to you. If I ask you to do this on a test, that will be given to you so that you can read it. And that is going to do it. So don't forget to go back to your Kia and to write a summary of this and to complete the attached worksheet. And I will talk to you again on the 30th. All right, gentlemen. And don't forget, your test is going to be on April 2nd. All right, so after the next lesson, April 2nd will be your test. These are all scheduled to go out on these days. All right, so have a good night. And if you have any questions, community periods, Mondays and Thursdays, 12:30 to 1:15. Thanks. Good night.